where does a thought actually become a physical reality? And I don't mean that metaphorically. I mean, at what physical point in the body does something abstract, like, say, the feeling of stress or fear, or even just cold, where does that transmute into an actual chemical that changes your heart rate or makes you shiver?
It's funny, that sounds like a question for a philosophy class, right?
Yeah,
But physiology has a very, very specific anatomical answer for that.
exactly. And that is where we're going today. For this deep dive, we are going right to the center of the brain. We're looking at the hypothalamus. And if you've ever just glanced at an anatomy textbook, you know, this thing it's tiny. It looks completely underwhelming.
It really does.
But then you start reading the source material. And for this, we are pulling heavily from two absolute cornerstones, Ganong's Review of Medical Physiology and the classic Guyton Hall textbook of medical physiology. And you realize this little 4-gram blob of tissue is basically running the entire show.
It is the ultimate interface. That is probably the best way to frame it from the get-go. It's the physiological bridge where the nervous system literally shakes hands with the endocrine system. It's the place where, as you said, mind becomes meat.
I love that image. And our mission today really is to map out this control room.
Right.
We're not just going to, list off a bunch of Latin names for nuclei because, frankly, without context, that's just a memory test and it's not very helpful.
Right. It's useless.
We want to understand the logic of the system. How does this tiny structure use the pituitary gland to maintain total body homeostasis? How does it integrate things like timing and feedback and raw survival drives?
And you mentioned the size, which is 4 grams. I think we need to just sit with that scale paradox for a moment because it's staggering.
It's absurdly small for what it does.
It is less than 1% of the brain's mass. I mean, Guyton makes this point very clearly. You could hold it on the tip of your pinky finger.
Yeah.
And yet, if you were to sustain damage, even minor damage to this tiny area, the consequences are immediate and they are catastrophic.
It's not like you just lose one specific skill.
No, you don't just lose a memory or the ability to do calculus. You lose the ability to regulate your body temperature. You lose the drive to drink water when you're dehydrated. You lose your appetite. Or on the flip side, you become unable to stop eating. You lose your sex drive. You lose your entire sleep-wake cycle.
The whole machine basically just stops running. The fundamental operating system crashes.
Precisely. The famous physiologist Sherrington, he called it the head ganglion of the autonomic system.
And that is such a great way to think about it. It isn't just a simple relay station. It's the CEO of your entire bodily maintenance program.
Okay, so let's orient ourselves. Let's get the lay of the land. If we were to, slice the brain open and look to this command center, where exactly are we pointing?
We're going deep. Right into the center. We're in a region called the diencephalon, and it's sitting right below the thalamus, which makes sense. That's where the name comes from. Hypothalamus.
Below the thalamus. Right. But the most critical piece of real estate, from a functional perspective, is what it sits directly on top of. It is positioned immediately above the pituitary gland.
And that proximity is no accident, I assume. They are physically tethered together.
Inseparable. You absolutely cannot understand hypothalamic function without understanding the pituitary. And the pituitary, it sits in this little protected bony pocket in the base of the skull called the sella turcica.
Which means Turkish saddle.
Turkish saddle, exactly.
Yeah.
It's a very, very protected spot. And the hypothalamus connects down to it via a little stalk called the infundibulum. Now, this is where we have to really dig into what we're calling the hardware, because the way the hypothalamus talks to the pituitary, it isn't uniform. It's actually a tale of 2 completely separate lobes.
Right. The sources make a massive distinction here between the posterior pituitary and the anterior pituitary. And looking at a diagram, you'd think they're just 2 sides of the same coin, but embryologically, you said it, they are completely different species.
That is the first major concept that you have to lock in. It's foundational. Let's start with the posterior pituitary, which is also called the neurohypophysis.
Neuro as a nerve.
Exactly. Because embryologically, this is actually an outgrowth of the brain itself. It is neural tissue. It's effectively the hypothalamus just extending itself downward like a drip of candle wax right into that saddle.
That's a direct connection.
Literally hardwired. You have these very large nerve cell bodies, these neurons, sitting up in the hypothalamus, specifically in 2 nuclei called the supraoptic and the paraventricular nuclei, and they send their long axons all the way down that stalk to terminate in the posterior pituitary.
Okay, so when we talk about the hormones that come from the posterior pituitary, They aren't actually made there. Is that right?
That's exactly right. The factory is upstairs in the hypothalamus. The hormones themselves are synthesized in the neuron cell bodies, then they're packaged into little vesicles, and they get shipped down the axons like a microscopic cargo train.
And they just wait there.
They sit there in the nerve endings at the bottom, just waiting for the signal. When an electrical signal, an action potential, fires down that nerve from the hypothalamus, they are released directly from the nerve ending into the bloodstream.
Genong has a specific term for this. He calls it neurosecretion.
Yes. And it's so important because it breaks the usual rule of neuroscience, where neurons talk to other neurons across a synapse. Here you have neurons talking directly to the bloodstream. It's a direct one-to-one conversion of electrical frequency into chemical concentration in the blood. Incredibly fast.
Okay, so that's the posterior lobe, the direct hardwired line. Now let's contrast that with the anterior lobe, the adenohypophysis.
The anterior lobe is a totally different beast.
Yeah.
It is not brain tissue. That's the key. Embryologically, it comes from something called Rathke's pouch.
Which, in plain English, is...
It's an invagination of the pharyngeal epithelium. Basically, it comes from the roof of your mouth. During development, this little bit of glandular tissue migrated upwards and just sort of stuck itself onto the front of the pituitary stalk. It's an immigrant, not a native.
So if it's not brain tissue, there are no wires, no axons connecting the hypothalamus down to the anterior pituitary.
Exactly right. No direct neural connection whatsoever. So the hypothalamus, the CEO, has a huge logistical problem. How does it give orders to the factory downstairs if there are no phone lines?
It must need some kind of courier system.
It needs plumbing. And this is, I think, one of the most elegant engineering solutions in the entire human body, the hypothalamic hypophyseal portal system.
That name sounds like something out of science fiction, but it's essentially just a private driveway for blood, isn't it?
That is a fantastic analogy. A private driveway. Think about it this way. If the hypothalamus just released its regulatory hormones into the general bloodstream, into the main vena cava, they would be diluted instantly by 5 liters of blood.
Like putting a single drop of food coloring in an Olympic swimming pool.
Exactly. By the time that blood circled all the way back around to the pituitary, the concentration of the hormone would be so low it would be completely undetectable. The signal would be lost.
So it would be useless.
Totally useless. So instead, the hypothalamus has this clever workaround. It releases its releasing hormones into a tiny specialized bed of capillaries right at the top of the stalk in a place called the median eminence.
Okay.
Now, these capillaries don't drain into the general body. They coalesce into long portal vessels that run straight down the stalk and then open up into a second capillary bed directly inside the anterior pituitary.
So it's a closed loop. It's a direct mail system from the boss's desk to the manager's inbox.
It is a mechanism purely for concentration. It allows the hypothalamus to secrete these minute nanogram or even picogram quantities of a hormone and yet deliver them at an incredibly high strength directly to the target cells. It's efficient, it's precise, and it keeps these very potent signals out of the general circulation where they might cause all sorts of unintended side effects.
Okay, so let's just summarize the hardware before we move on to the software. The posterior pituitary is a neural extension. It's all about wires and electricity. It's fast. The anterior pituitary is a separate gland connected by a private blood vessel system. It's all about plumbing and chemistry.
That is the fundamental architectural split. And once you see that clearly, the software, the hormones themselves, and how they're controlled starts to make a whole lot more sense.
All right, let's dive into that software. Let's start with the hardwired side, the posterior pituitary, the direct line. We're dealing with 2 main hormones here, right? Vasopressin and oxytocin.
Yes. Let's take vasopressin first. You'll also see it called ADH, which stands for antidiuretic hormone. And that name tells you exactly what it does. It is, in essence, the body's water warden.
Its job is to prevent diuresis, to keep water inside the body.
Precisely. Let's create a scenario. Imagine you're dehydrated. Maybe you're on a long hike in the sun, you're sweating a lot, and you just ran out of water. 2 things are happening in your blood. Your blood volume is dropping, and the concentration of salt and other solutes is rising.
It's becoming hypertonic.
Exactly. Now, the hypothalamus contains these amazing cells called osmoreceptors. They are specialized cells that literally shrink when the fluid around them gets too salty. Water gets pulled out of them by osmosis.
And that physical shrinking acts like a trigger.
That's the signal. They fire action potentials. They're basically screaming, "Dehydration! We're losing water!" And that electrical signal travels down those long axons from the hypothalamus to the posterior pituitary, causing the nerve endings to dump ADH into the blood.
Okay, so now ADH is in the circulation. Where does it go?
It travels to the kidneys. Specifically, it targets the very last part of the kidney tubules, the collecting ducts.
And normally those ducts are like waterproof pipes, correct? The
Ah.
water in the pre-urine just flows right through them and eventually becomes urine?
Correct. They are impermeable to water. But ADH initiates this brilliant cellular maneuver. It binds to receptors on the kidney cells and tells them to take these little vesicles. Think of them as tiny bubbles inside the cell that are preloaded with water channels called aquaporins and fuse them to the cell membrane.
So it physically installs plumbing into the wall of the pipe.
It punches holes in the barrier. It's like opening up a series of floodgates. And suddenly, all that water that was destined for the toilet rushes out of the duct and back into your bloodstream. You save the water, your blood volume is restored, and as a result, your urine becomes very dark and very concentrated.
And then what happens when you finally get to a stream and drink a big glass of water?
The reverse process. Your blood becomes more dilute, the osmoreceptors swell back up to their normal size, they stop firing, the ADH signal stops, and the kidney cells pull all those aquaporins back inside. The wall becomes waterproof again. It's an incredibly dynamic and responsive system.
And we can really see what happens when this system breaks. The notes mention the clinical condition diabetes insipidus.
Yes, and it's absolutely crucial to distinguish this from diabetes mellitus, the one everyone knows as sugar diabetes. The word diabetes just refers to a siphon, the excessive urination.
Right, but mellitus means sweet, like honey. And insipidus means tasteless.
It literally means tasteless.
Hmm?
And that refers to the taste of the urine. It sounds grim, but before modern labs, physicians would actually taste a patient's urine to help with diagnosis.
A very brave profession.
Indeed. In diabetes insipidus, the problem isn't sugar. The problem's water. You either can't make ADH in your brain, which is called central DI, or your kidneys have become deaf to its signal, which is nephrogenic DI.
So you can't install the aquaporins?
You can't install the plumbing. You cannot save water. So these patients, they pass incredible volumes, liters and liters, sometimes up to 20 liters a day of clear, dilute urine. They live in a state of frantic, unquenchable thirst because they're essentially leaking water just as fast as they can possibly drink it.
It paints a very clear picture of just how vital that one hormone is. Okay, so that's ADH. The other neural hormone is oxytocin. Now, the popular press absolutely loves to call this the cuddle hormone or the love molecule.
Which is, it's partially true. It definitely plays a role in social bonding and trust. But from a purely physiological standpoint, oxytocin is much more aggressive. It's about muscle contraction, specifically smooth muscle.
Where specifically are we talking about?
Two main places with two very powerful actions. The first is the uterus during labor, and the second is the breast during lactation.
Let's talk about lactation first, because the letdown reflex is the absolute textbook example of this direct neural loop we've been describing.
It is. It's a perfect neurohormonal reflex. So just visualize the process. A baby suckles at the breast. That tactile sensation, the simple touch on the nipple, sends a neural signal up the spinal cord directly to the hypothalamus.
The hypothalamus receives the data point. Infant is attempting to feed.
And in response, it fires those oxytocin-producing neurons. The hormone travels down the axons to the posterior pituitary, gets dumped into the blood, and travels systemically until it reaches the breast.
And what does it do there?
In the breast, it targets a special kind of cell called a myoepithelial cell. These are muscle-like cells that wrap around the tiny milk-producing glands, the alveoli, like a net or a mesh bag.
And oxytocin makes them clench, like squeezing a sponge.
It makes them contract powerfully and literally squeezes the milk that's been produced out of the glands and into the ducts so the baby can get it. This is why it's called milk ejection, not milk production. It's an active process.
And this is exquisitely sensitive to the mother's environment, right? The notes mention that stress can actually block this reflex.
Absolutely. This is where the hypothalamus really shows its role as an integrator. If the mother is terrified or in pain or extremely stressed, the sympathetic nervous system, the fight-or-flight system, kicks in and can inhibit the release of oxytocin from the hypothalamus. The milk just won't let down.
From an evolutionary perspective, that makes sense. It's not safe to feed right now. Danger is present.
Exactly. And speaking of survival mechanisms, Guyton points out a fascinating quirk about oxytocin's other major role in labor. Most physiological systems in the body run on negative feedback. The logic is, I have enough of something, so please stop making it.
Right, it's self-regulating. It keeps things stable.
But oxytocin in labor is one of the very rare and very important examples of positive feedback.
The snowball effect, the chain reaction.
Precisely. It works like this. The baby's head begins to push against the cervix, stretching it. That stretch sends a neural signal up to the hypothalamus, which says, release oxytocin. Oxytocin travels to the uterus and causes it to contract,
Which pushes the baby harder against the cervix.
which causes more stretch, which triggers the release of more oxytocin, which causes stronger and more frequent contractions. It's a self-amplifying loop that is designed not for stability, but for an explosive, culminating event, the birth of the baby. It only stops when the stimulus, the stretching of the cervix, is removed.
Incredible. Okay, so that covers the hardwired posterior side. Let's pivot now to the middle management side, the anterior pituitary, the side that's connected by that special portal blood vessel system.
Right. And this is where we see the classic endocrine hierarchy. If the hypothalamus is the CEO in the penthouse office, the anterior pituitary is the vice president operations on the floor below. The CEO doesn't talk to the factory floor workers directly.
It sends memos, the releasing hormones and inhibiting hormones.
It sends memos down to the VP, and the VP reads the memo and then issues specific directives to the different departments, the specific organs like the thyroid, the adrenals, the gonads.
So let's walk through a few of the main axes. The thyroid axis is a classic.
A perfect example. The hypothalamus senses, let's say, a drop in your core body temperature or a fall in your overall metabolic rate. It sends out a memo called TRH, thyrotropin-releasing hormone.
That memo travels down the private driveway, the portal system.
Reaches the anterior pituitary. The specific cells there that have TRH receptors read the memo, and in response, they release their own hormone, TSH, thyroid-stimulating hormone, into the general circulation.
And TSH then travels to the thyroid gland in the neck.
Right, where it tells the thyroid to pump out thyroxine, or thyroid hormone, which then goes to all the cells in the body and revs up the metabolism to generate more heat.
And there's a feedback loop here too, I assume, to turn it off.
Yes, and this one is a classic negative feedback loop. High levels of thyroxine in the blood go back up to both the pituitary and the hypothalamus and essentially say, okay, mission accomplished, we're warm enough, you can slow down now. It inhibits the release of both TSH and TRH.
And we see this same three-tiered structure for the stress response, right?
The HPA axis. Exactly the same logic. Hypothalamus releases CRH, corticotropin-releasing hormone. The pituitary releases ACTH, adrenocorticotropic hormone, and the adrenal glands release the stress hormone, cortisol. Cortisol then feeds back to shut the system down.
And for reproduction.
Same pattern. The hypothalamus releases GnRH, gonadotropin-releasing hormone. This tells the pituitary to release two hormones, LH and FSH, which then go to the gonads to regulate the menstrual cycle or sperm production.
Now, there's one oddball in this group that the texts highlight. Most of these hypothalamic signals are go signals, release this, stimulate that. But there's one that is primarily a stop signal.
Yes, and it's a very important one. That's dopamine. In the context of the pituitary, we often give it a different name, PIH, which stands for prolactin inhibiting hormone.
So its main job is to constantly hold back the release of prolactin.
That's exactly right. The default state of the cells in the anterior pituitary that make prolactin is to just make it.
Yeah.
They are tonically active. So the hypothalamus has to keep a foot on the brake by constantly dripping dopamine onto them via those portal vessels.
So what would happen if you severed the pituitary stalk? If you physically cut the connection between the brain and the gland?
That's a classic thought experiment in physiology, and it reveals the logic beautifully. All the other hormones we mentioned, thyroid, adrenal, reproductive, their levels would crash to near zero because they're no longer getting their go signals, the releasing hormones. But prolactin,
It would skyrocket.
it would skyrocket.
Yeah.
Because you just cut the brake line, and that's a crucial diagnostic clue for clinicians. If you see a patient with low thyroid, low cortisol, and low sex hormones, but a very high prolactin level, you have to think about something compressing or damaging that pituitary stalk, like a tumor.
Before we move on from this section, I want to touch on a clinical story from Genong that I just found fascinating. Kallmann syndrome. It reads like a detective story about a missing roadmap.
Oh, Kallmann syndrome is a beautiful, if tragic, example of how embryology dictates physiology. Patients with this syndrome, who are usually males, present with two seemingly unrelated problems. The first is hypogonadism. They never go through puberty.
Yeah, a clear hormonal issue.
Right. And the second is anosmia. They have no sense of smell.
Which seems like a totally random combination. I mean, what on earth does the nose have to do with the testicles?
That was the mystery for decades. And the answer wasn't found in hormones, but in tracing the development of the neurons themselves. It turns out that the specific neurons that are responsible for making GnRH, the puberty starter signal, they don't actually originate in the brain.
Wait, what? Where did they start?
They start in the nose. In the embryonic olfactory plight code, the structure that will become the sense of smell. And as the fetus develops, these neurons have to physically migrate. They crawl up along the olfactory nerve fibers, pass through a bony plate in the skull called the cribriform plate, and find their way to their final home in the hypothalamus.
That sounds like a ridiculously dangerous commute for such important cells.
It is an incredibly perilous journey. And in Kalman syndrome, the adhesion molecules that guide them, the molecular roadmap, is defective. The neurons get lost, they never complete the migration, they never make it to the hypothalamus.
So the patient has no sense of smell because the olfactory tract itself is malformed, and they have no puberty because the GnRH neurons that are supposed to kick everything off never arrived at their destination.
Exactly. It's a perfect, poignant illustration that the hypothalamus isn't just a monolithic block of tissue. It's a carefully orchestrated assembly of parts that have to find each other during development. Structure dictates function.
Let's zoom in on one specific hormone from the anterior pituitary now, because the note suggests it deserves its own deep dive within our deep dive. That's growth hormone, or GH.
Yes, the master builder.
Yeah.
Or maybe the master metabolic strategist.
Why does this one get special treatment? You know, TSH just talks to the thyroid. ACTH just talks to the adrenals. Who does GH talk to?
Everybody. That's the difference. It exerts significant effects on almost all tissues in the body. And while we always associate it with children growing taller, its role in adult metabolism is arguably just as critical. It's a key player in how we manage our energy stores.
Let's talk about that strategy. The outline mentions a specific set of trade-offs that GH orchestrates, particularly during a state like starvation.
Okay, let's think about this from an evolutionary perspective. If you are starving, you have a critical energy crisis. You desperately need fuel to stay alive. But you have a problem. If you start burning your muscles for fuel, which is high-quality protein, you become weak. If you're too weak to hunt or gather food or escape a predator, you die.
So the number one priority is to preserve your muscle mass at all costs.
Enter growth hormone. When you are fasting or starving, GH levels spike dramatically. And it delivers a very clear three-part metabolic message to the body. Part 1. It is strongly anabolic for protein. It tells the muscle cells, do not break down, hold on to your amino acids, save the structure.
Okay, so preserve muscle. Got it.
Part 2. It is strongly ketogenic and lipolytic. It tells the fat cells, the adipocytes, mobilize, break down your triglycerides and dump fatty acids into the blood. We are going to burn the long-term energy reserves.
So it switches the body's primary fuel source from protein to fat in order to save the muscle.
Correct. But there's a third, slightly counterintuitive part to its message. It tells most of the cells in the body, stop using glucose. It actively decreases carbohydrate utilization by peripheral tissues. We call this a diabetogenic effect because, as a result, it raises blood sugar levels.
Okay, why would it do that? In a state of starvation, why would you want to stop the body from using a high-quality fuel like sugar?
To save it for one incredibly picky customer, the brain. The brain runs almost exclusively on glucose or on ketones in a real pinch. So GH orchestrates this beautiful metabolic rationing. Okay, everyone, muscles, you eat the fat. Brain, we get the limited supply of sugar. And nobody touched the protein structure until it's an absolute last resort.
That is an incredibly sophisticated survival strategy. It's not just growth. It's about resource allocation under pressure.
It is. Now, for the actual growth part, the part that makes bones longer, GH acts a bit like an architect. It draws up the plans, but it doesn't lay the bricks itself. It goes to the liver and orders the production of a group of contractors called IGFs, or insulin-like growth factors. The main one is IGF-1.
Which you'll also see called somatomidin C in older texts.
Right, and it's this IGF-1 that actually travels to the epiphyseal growth plates of the long bones and stimulates the cartilage to proliferate, which is what makes you taller.
And this brings us to the pygmy paradox that was mentioned in the text, which really drives this point home.
Yes, this was a fascinating discovery. Scientists studying African pygmy populations were trying to understand the genetic basis for their short stature. They naturally assumed they would find very low levels of growth hormone. But when they measured it, they were shocked. The GH levels were normal, sometimes even higher than average.
So the architect was on site screaming, build, build, build.
But the building wasn't happening. And the reason was that this population has a genetic variance where their livers don't produce significant amounts of IGF-1 in response to GH. The contractor was missing from the job site.
It really highlights that you need the entire chain of command to be intact. Signal, mediator, and effect. Just having the hormone isn't enough.
It's a perfect illustration of that principle.
What about timing? When are we actually making this stuff? Is it just a steady trickle?
Not at all. It's highly pulsatile. It comes in bursts. And the single biggest, most massive spike of your entire daily production happens during the first 2 hours of deep, slow-wave sleep.
So the old saying about needing growing sleep is a literal biological fact.
It is an absolute fact.
Yeah.
If a child has chronically fractured sleep, due to something like sleep apnea, for instance, they may not reach their full growth potential because they miss that critical window. And here's the kicker for all of us. As we age, that nocturnal spike gets progressively smaller and smaller. An elderly person produces maybe only 25% of the GH that a teenager does.
Which would go a long way to explaining the age-related loss of muscle mass, the frailty, the increase in body fat that we see.
It is a major, major driver of the aging phenotype, no question.
Let's talk about what happens when this system goes haywire. The texts draw a very sharp contrast between gigantism and acromegaly. It's the same hormone, the same excess production, but the result looks completely different depending on when it happens.
Yes, and the entire difference hinges on one thing: the state of the growth plates in the bones. When you are a child or an adolescent, the ends of your long bones, the epiphyses, are still open. They have a layer of cartilage that can expand and create new bone.
So if you have a pituitary tumor that's pumping out massive amounts of GH then.
You stretch, you get gigantism. The skeleton just keeps lengthening and lengthening. You can become 7-8 feet tall.
Like Andre the Giant, he's a famous example.
A classic example of pituitary gigantism. But what happens if that same tumor starts after you've gone through puberty, as an adult? By then, your growth plates have fused. They have turned to solid bone. They cannot get any longer.
So where does all that growth signal go? It has to go somewhere.
It goes out, the bones get thicker, the soft tissues expand. This is acromegaly. The word literally means enlarged extremities.
And it's a much more disfiguring condition from the descriptions.
It is. The hands and feet become huge and spade-like. The jaw protrudes, creating that classic lantern jaw appearance. The brow ridges thicken, the tongue can enlarge, even the vertebrae thicken, leading to a hunchback or kyphosis. It's a dramatic change in appearance.
It's just amazing that timing alone, that one single variable of whether the plates are fused or not, dictates whether you become a proportional giant or develop the features of acromegaly.
Physiology is all about context. The signal is the same. The state of the receiver has changed.
Context, that is the perfect transition to the next section of our deep dive. Because the hypothalamus isn't just blindly sending out these hormonal orders, it is constantly reading the context of the entire body. It is the great integrative center.
This is the housekeeping logic. How do we keep the whole machine running smoothly?
Let's start with the big one, temperature. The body's thermostat.
And it functions almost exactly like the HVAC system in your house. The hypothalamus has a set point, which is usually around 37 degrees Celsius or 98.6 Fahrenheit. And it has two distinct centers to manage this. The anterior hypothalamus acts as the body's air conditioner. It contains heat-sensitive neurons.
And if the blood flowing through it is too hot?
It triggers cooling mechanisms. First, vasodilation. Your skin flushes red as blood is shunted to the surface to dump heat into the air. And if that's not enough, sweating, which cools you by evaporation.
And the posterior hypothalamus.
That's the eater. It contains cold, sensitive neurons. If your blood temperature drops, it triggers shivering, which is rapid involuntary muscle contraction to generate heat through friction, and vasoconstriction, where your skin goes pale to keep all the warm blood in the core away from the surface.
But what about fever? I think most people, myself included, tend to view a fever as the thermostat breaking. My system is failing, it's overheating.
That's a common misconception. A fever is not a broken thermostat. It's a thermostat that has been deliberately reprogrammed to a higher setting. It is an active, adaptive defense strategy.
Walk us through that mechanism. How does an infection tell the brain to turn up the heat?
Okay, so you get an infection, say a virus or bacteria. Your immune cells, your white blood cells, respond by releasing inflammatory chemicals called cytokines, things like interleukin-1 and TNF-alpha. These chemical signals travel through the blood to the brain, and they hit a specific spot called the OVLT.
The organum vasculosum of the lamina terminalis?
A mouthful, I know, but it's an important spot. It's one of the few places where the blood-brain barrier is intentionally leaky. This allows the hypothalamus to taste the blood for these cytokines. When it detects them, it says, aha, we're under attack. Most bacteria and viruses replicate best at 37 degrees. Let's make the environment hostile. Let's cook them out, crank the thermostat to 39 or 40 degrees.
So the internal set point physically moves up?
The target is now 39. And this immediately explains why you get the chills when a fever is starting.
Right. I've always wondered that. Why do I feel freezing cold and start shivering when my temperature is actually rising?
Because your actual body temp is still 37, but the boss in the hypothalamus says it should be 39. The hypothalamus compares the two numbers and says, emergency, it's freezing in here. We are 2 degrees below our target. So it triggers the heating mechanisms. It makes you shiver. It makes you curl up under a blanket.
It forces you to generate and conserve heat until your body temperature matches the new higher setting.
Exactly. And then, when the fever breaks, when your immune system wins, the cytokines go away and the set point drops back to 37, you suddenly feel incredibly hot. You start sweating profusely, you throw off all the covers. Because now your body's at 39, but the target is back down to 37, so you have to dump all that excess heat.
That is such a clear way of explaining it. It's all relative to the setting on the dial.
Precisely.
Okay, let's look at thirst, another one of these primal integrative drives. How does the hypothalamus know that we need water?
It tastes the blood.
Mhm.
Literally.
We mentioned the OVLT for fever, but there's another spot involved here too, right? The subfornical organ.
Yes. These are two of the key circumventricular organs. They sit outside the full protection of the blood-brain barrier, so they are constantly sampling the chemistry of your blood. And they are looking for two main signals for thirst.
What are they?
The first and most sensitive is osmolality. How salty is the blood? We talked about this with ADH. If you eat a bag of salty pretzels, your blood gets more concentrated. The cells in these organs physically shrivel up as water leaves them by osmosis. That shriveling is the direct trigger for the sensation of thirst.
And what's the second trigger?
Low volume. This is for more dramatic situations like hemorrhaging or severe dehydration. Your blood pressure drops. In response, your kidneys release a hormone called angiotensin II into the blood. The subfornical organ has receptors that detected angiotensin II, and this triggers an incredibly powerful urgent thirst. It's a chemical alarm system for low blood volume.
So it's both a concentration sensor and a volume sensor.
And a rigorous one. It's a drive you can't ignore. It will override almost everything else.
Okay, one last major maintenance function. Time. The circadian rhythm.
The master clock. This is the suprachiasmatic nucleus, or SCN, a tiny pair of nuclei sitting right on top of the optic chiasm.
And how does a small blob of cells actually keep time? Is it like a tiny hourglass in there?
It's a molecular feedback loop. This is Nobel Prize-winning science. It's that elegant. Inside each of the neurons of the SCN, you have a set of clock genes with fantastic names like CLOCK and BML1.
I do appreciate a clear naming convention.
These genes turn on and start producing their respective proteins. These proteins then pair up in the cell cytoplasm, and they start to accumulate and accumulate over a period of many hours.
And then what happens?
Once they reach a critical mass, they're able to loop back into the nucleus, and they physically inhibit their own production. They shut down the factory that made them. After that, the protein levels slowly degrade and crash. When they get low enough, the inhibition is lifted, and the genes turn back on, starting the whole cycle over again.
And that entire cycle buildup, shutdown, crash, and restart takes how long?
Approximately 24 hours. It is a beautiful, self-sustaining molecular oscillator.
But it can't be perfect, right? It must need to be synchronized with the real world every day, otherwise we drift out of phase.
That's the process of entrainment. And this is where the SCN's location is so key. It sits right above the optic chiasm, where the optic nerves cross. It gets a direct feed from the retina via a pathway called the retinohypothalamic tract. Sunlight hits specialized cells in your eye. That signal goes straight to the SCN, and it resets the protein cycle every single morning.
The sun is up, let's restart the clock.
Exactly. And this is why jet lag feels so awful. Your internal molecular protein cycle is still on New York time, but the sun is telling you it's Tokyo time. It takes several days for your chemistry to painfully realign itself with the new light-dark cycle.
Okay, we've covered the machine, the hormones, the thermostat, the clock, but there is one final crucial piece of the puzzle that takes the hypothalamus from being just a machine to being a creature, and that is the limbic connection.
Emotion is physiology. This is my favorite part because it connects everything.
The outline calls this the border system because the hypothalamus isn't just an isolated controller. It's the primary output mechanism for the entire limbic system, the emotional brain.
Right. You have to realize that you cannot have an emotion without a physical response. It's impossible. If you feel fear, your heart must race. If you feel rage, your muscles must tense. If you feel pleasure, your physiology changes. The hypothalamus is the structure that makes that happen. It translates the feeling into a physical reality.
The notes from Guyton discuss very specific reward and punishment centers. This isn't abstract psychology. This is literal wiring.
It is hardwiring. If you place an electrode into the punishment center, which is located mostly in the central gray area around the aqueduct of Sylvius, and you stimulate it in an animal, it doesn't just look a little annoyed. It shows signs of pure terror.
Yeah.
It will hiss, spit, and try desperately to escape. It's experiencing pain and fear.
And the reward center.
That's located along a pathway called the medial forebrain bundle. Stimulation there brings about intense placidity, tameness, and satisfaction. In those classic experiments, you'll see a rat press a lever to self-stimulate this area thousands of times an hour. They will ignore food, water, even mates, just to keep pressing that pleasure button until they collapse from exhaustion.
But there's a hierarchy here, isn't there? One is stronger than the other.
Yes, and it's a critical one for survival. Punishment trumps reward. If you stimulate the fear center, it completely inhibits the pleasure center.
Which makes perfect evolutionary sense.
Completely. If you were enjoying a delicious meal, reward, and a tiger walks into the room, fear, you need to stop eating instantly and run. If pleasure overrode fear, you'd become dinner. Fear has to have veto power.
Speaking of eating, the connection between hunger and rage is explicitly laid out in the texts. The modern term hanger, being hungry and angry, is real physiology.
It is very, very real. The lateral hypothalamus is known as the hunger center. If you stimulate it, an animal will eat voraciously. But if you increase the intensity of that stimulation, it triggers overt rage, full-on fighting behavior.
So the fundamental drive to acquire food and the drive to fight are wired right into the same bundle of nerves.
They are next-door neighbors. And conversely, the ventromedial nucleus is the satiety center. Stimulating it causes an animal to stop eating and become very tranquil. But if you create a lesion in that center, if you destroy it.
The animal never feels full, right? It becomes obese.
Massively obese, yes. That's the classic finding. But what's often overlooked is that it also becomes savagely vicious. It loses its sense of calm. It suggests that a significant part of our personality, whether we are calm or aggressive, is tied to the delicate balance of the firing rates in these tiny, tiny nuclei.
Finally, we have to talk about the amygdala. Guyton calls it the window of the limbic system. What does he mean by that?
The amygdala is the great assessor. It takes in all the complex sensory data from the world, what you're seeing, what you're hearing, what you're smelling, and it asks a series of rapid, primitive questions. Is this dangerous? Can I eat this? Can I mate with this?
And then it sends its assessment to the hypothalamus, which then executes the appropriate response.
Precisely. The amygdala provides the intelligence and the context. The hypothalamus provides the raw execution. And the classic example of what happens when you lose this window is Kluver-Bucy syndrome.
The monkey experiments from the 1930s?
Yes. Researchers surgically remove the anterior temporal lobes in monkeys, a procedure which includes the amygdala. The monkeys' personalities changed instantly and profoundly. First, they lost all sense of fear. They would approach and try to pick up snakes.
Because they could no longer assess the context of danger.
Right. They also became hypersexual, attempting to meet with other animals, inanimate objects, anything. And they developed what was called psychic blindness. They would put dangerous objects like lit matches or nails into their mouths because they could no longer process the visual context of this is not food or this will harm me.
So they still had all the basic hypothalamic drives, eat, mate, explore, but they had no filter, no window to look through to make sense of the world.
Exactly. It shows that the hypothalamus provides the engine, but the amygdala provides the steering. You need both.
So let's step all the way back. We have this tiny 4-gram controller that is managing our water, our heat, our growth, our sleep, and the most basic drives of fear, rage, hunger, and pleasure. What is the big overarching takeaway here?
For me, the deepest lesson from the hypothalamus is about the nature of control itself. We tend to think of control as being about giving orders, about being dominant. But the hypothalamus teaches us that true effective control is about awareness. The hypothalamus almost never acts without listening first. It is constantly tasting the blood for salt and sugar. It's listening to the baroreceptors in the heart for pressure. It's checking the temperature sensors in the skin and the core. It's reading the clock genes to know the time of day. It's consulting with the amygdala about the emotional state of the world. It integrates 1,000 different data points before it releases a single drop of a single hormone.
Control emerges not from dominance, but from contextual awareness.
Precisely. It is not about dominance. It is about synthesis. And that is a powerful lesson.
There's one final thought from the notes, a concept from Guyton on how the brain learns that I really want to leave our listeners with. It's the idea of habituation versus reinforcement.
This is a profound point to end on, I think. Guyton notes that the brain is just bombarded with sensory information every single second. The feel of your clothes on your skin, the hum of the refrigerator, the background traffic noise. The brain has to ignore 99.9% of it. It habituates.
We just delete the noise so we can function.
But if a signal causes either pleasure or pain, if it activates the limbic system, the reward or punishment centers, the brain says, whoa, pay attention, save this. That is reinforcement.
So what you're saying is we literally only learn what we feel.
Essentially, yes. Information without an emotional tag is treated as irrelevant and is discarded. If you want to remember something, or if you want to teach someone something effectively, you have to attach it to a feeling. The fundamental physiology of the limbic hypothalamic system demands it.
I think that's a perfect and slightly mind-bending place to stop. We've certainly felt the importance of this topic today.
Absolutely. It's the center of everything.
If you want to see the actual wiring diagrams and the feedback loops, and they are really beautiful in their complexity, we strongly encourage you to check out the relevant chapters in Genome, that's the chapter on hypothalamic regulation, and in Guyton and Hall as well.
Highly recommended. Just seeing the diagram of the portal system alone is worth the price of admission. It makes it all click.
Thanks for diving deep with us. We'll catch you on the next one.